So we've known about the existence of ctDNA, circulating tumor DNA, for a while. And one of the key challenges is it makes up such a small amount of the total cell-free DNA in the blood. In order to detect it, you really have to be very sensitive and very specific from an assay perspective. We've used so-called tumor-informed approaches, where you take a biopsy specimen, you sequence that specimen, identify mutations, and then you search for them in the blood. And using increasing numbers of mutations has led to better sensitivity.
Well, so what's remarkable about NeXT Personal is its ability to detect traces of circulating tumor DNA down to this 1 part per million level. We take whole genome sequencing, we develop this NeXT platform technology, and we combine those to be able to use that to create a personalized signature of the patient's tumor. This is a personalized approach for every patient. So that signature, once we develop it for each patient, we can actually look for that signature in the blood samples that we get from the patient to detect the tumor at that 1 part per million sensitivity level. And we demonstrated that with TRACERx, and we showed that that level of sensitivity is incredibly powerful and really can change management and hopefully outcomes for those patients.
And so I think one of the key questions that kind of was the setting for this work was, what are the clinical implications of ever increasing sensitivity? What we wanted to test in our most recent work was, how much could we improve the clinical sensitivity of ctDNA as a biomarker for low-risk disease using an ultra-sensitive assay? What we discovered was that patients with ctDNA present at ultra-low levels behave markedly differently to those where we weren't able to detect ctDNA at all. So really suggesting that an ultra-sensitive assay could be an important biomarker for low-risk disease.
One of the exciting takeaways for me from the TRACERx study is that we demonstrated a pretty big leap forward in terms of ability to impact the clinical process all the way from pre-surgery all the way through post-surgery, during adjuvant treatment, and actually for recurrence monitoring. You know, that's a really exciting prospect, not for lung cancer patients, but for cancer patients in general.
The new data we put out a couple of days ago revealed that patients with ctDNA present preoperatively do markedly worse than those where we weren't able to detect it with our ultrasensitive assay. That opens up some interesting questions in terms of how we can really drive home this advantage to improve patient care. So what we were able to show in the TRACERx study was that we were able to identify quite crisply groups of patients with a favorable prognosis who potentially have lower risk disease and might be a candidate for de-escalation of therapy and surveillance, as well as patients with high risk stage 1 disease who might very well be a candidate for treatment escalation. And so I think that's one of the things we're really excited about, is thinking about implementing that in the context of prospective studies going forward.
One of the most important takeaways is showing that the ultra sensitivity makes a difference in lung cancer. And we can see that dramatically in the baseline pre-surgical data that we in TRACERx, where we have much higher detection rates for especially adenocarcinoma, stage 1, 2, and 3 than had been previously reported, which is critical for this to be something that could be useful at the bedside for lung cancer patients.